A pleasant day to all. First, I'd like to thank the organizers of this big event and MiRXES for giving me the opportunity to share our learnings, the memorable and fun experiences, and the clinical validation of the Fortitude 2.1 COVID-19 RT-PCR test in the Philippines. When I accepted this clinical validation, I realized that as a scientist, I have been living in a bubble. At first, I expected that things would be a breeze. But doing research in a pandemic is totally a different story. I encountered several challenges that I did not encounter pre-pandemic. In the next slides, I will be discussing the different challenges my team has encountered during the implementation of the clinical validation, which I have categorized into the following: ethical approval stage, study site and study design, participant recruitment, safety of the team, and molecular analysis. When we first submitted our proposal to the Research Ethics Review Committee of our proposed study site, we were expecting for an expedited review since our research involved the study of diagnostic kit for COVID-19, and the research procedures presented no more than the minimal harm to the research participants or communities. We forgot to consider that the chair of the ethics committee is a COVID-19 frontliner of the hospital study site. There was ethics committee staff shortage due to exposure to COVID-19, and the lay reviewers preferred the printed copies of the documents over online review process. Hence, I had to send over the documents, further delaying our clearance. To top it all, the legal reviewer required the local partner to be involved in the research contract. Since our team is experienced in research that involve human samples, we already have templates for ethics review that we just modified to fit the current study. Hence, the contents of the proposal was approved after just one round of minor revision. We also patiently and constantly followed up while remaining sensitive to local realities. As MiRXES is familiar with local regulatory procedures, the legal reviewer's requirement was easily addressed. Way back in May 2021, we selected Ilocos Norte, a province in the northern Philippines, 512 kilometers from Manila, as our study site. And for the following reasons. It is a government-run COVID-19 referral facility with a Department of Health accredited COVID-19 testing lab. I have existing collaborations with them on my cancer studies. The Research Ethics Review Committee is accredited to review clinical trials, and most importantly, their COVID situation at the time of submission of this ethics proposal would not impede emergency response efforts. When we were about to send the equipment and materials to the study site, our research partners in the area could no longer commit because COVID-19 cases started to dramatically increase after detecting the Delta variant in the region, overwhelming now the facility. And there were limited staff to do the Fortitude 2.1 clinical validation on top of their normal duties. At this point, there was no turning back. I am accountable, and I was committed to successfully implement the trial despite all the odds. While waiting to get cleared by the ethics committee, I would regularly monitor the COVID-19 situation in the area, and I noticed that cases consistently increased each day. Hence, I started drafting contingency plans in case we change study site. Eventually, my instinct was right. When I presented my Plan B to MiRXES Director for Strategic Partnerships, she already had the same thing in mind. Finally, we were able to subcontract a private DOH-accredited COVID-19 molecular testing facility. We realized that it may not be wise to partner with government-run hospitals at this time. We had our kickoff on the 6th of August of this year. My team was confident that we would be able to complete the required 300 and 500 negative cases within just 2 weeks, since the Philippines during the time would record at least 20,000 cases a day. We thought that it would be effortless to invite potential participants to join the study and that everyone knew how to navigate the online forms, or at least they would be easy to train on the use of technology. In the past, it would be easy for my team to recruit cancer patients and clinically healthy controls to join our study. However, recruiting participants for this current study was a different story. Many hesitated to join because of stigma stemming from being infected with or having close contacts with COVID-19 cases. This photo shows a common scenario in the Philippines. A COVID-19 patient being picked up by an ambulance and surrounded by healthcare workers in their full PPE. The photos show our team in a community-based testing center and a typical quarantine facility in Metro Manila. We have observed that it was difficult to invite patients in quarantine facilities. The photos show our team in a community-based testing center and a typical quarantine facility in Metro Manila. We have observed that it was more difficult to invite patients in quarantine facilities than those who were to be tested for the first time. Some patients in isolation rooms complained of pain and discomfort related to swabbing. Moreover, we observed that daily wage earners, despite being close contacts and symptomatic, were hesitant to get tested because a positive result was equated to loss of income. So, you may know that the Philippines is an archipelago, but what you may not know is that the number could go up or down depending on whether it's high tide or low tide. So, it was really crucial for the team to know when to go to the study site and whether we would need a boat. In addition, the team had to learn to translate the study into different dialects. In fact, in one of the towns we went to, the residents had to translate the study to three different dialects. The Philippines has been cited as the social media capital of the world. Hence, we expected our participants to have their own email addresses and that they are knowledgeable or easy to train in the use of online forms, as it would be risky and waste of time to be explaining to each of them how to navigate the system. And the biggest challenge was getting access to the areas with high COVID-19 cases, getting access to quarantine facilities, and the community-based testing sites. We were faced with a lot of political and administrative hurdles then. So in the past, I would only need to present to the medical director and department heads of the hospital whenever I would invite for collaboration. But in this clinical trial, I was taught of the importance of understanding local power dynamics in order to access potential participants. I wrote governors, mayors, municipal health officers, held virtual meetings with them. I had to ask the support of medical technologists to have access to COVID-19 testing centers. I even approached factory workers and factory owners, offering to have them tested for free. I also invited healthcare workers, especially the volunteers in vaccination sites. Along the way, I learned to develop genuine collaboration with local stakeholders, becoming more sensitive to the needs of the community. As I mentioned earlier, daily wage earners would usually evade testing because of of income if they tested positive or identified as close contacts. Hence, One municipal health officer recommended to convert the monetary compensation to sack of rice per participant so that the family had something to eat while they are under quarantine. Surprisingly, close contacts started queuing in our booth, and we were able to recruit 310 participants from an area where the positivity rate at the time was at 60%. So this clinical trial became beneficial to the local community because the timely release of their official COVID-19 results helped identify and isolate the infected individuals. Our clinical validation experience is in line with MiRX's values. The Philippines is among the countries that benefited from MiRX's humanitarian efforts, especially when the pandemic just started and the Philippines had limited testing capacity then. To reach more volunteers and to have well-represented participants in terms of age, sex, socioeconomic status, we also use the social media as our recruitment tool. When we were starting, I was confident about the people I hired. All the drivers and swabbers were fully vaccinated. The swabbers have passed the biosafety training and certification exams, and they have never been infected when they worked as swabbers when the pandemic just started. As we picked up with our recruitment, it dawned on me that my drivers and swabbers are at a very high risk of getting infected with the virus. Eventually, all of them tested positive and had to undergo mandatory quarantine. Sad to say that all my swabbers resigned afterwards. So from the time this project started, I would always have contingency plans in mind. Days before my team got infected, I have been recruiting and training swabbers from high-risk sites. Thus, while my original team was undergoing mandatory quarantine, I was able to continue with participant recruitment. I became more stringent in implementing biosafety guidelines, and everyone had to undergo weekly COVID-19 testing to ensure their safety and the people that they swabbed. In this clinical validation, the nasopharyngeal samples were collected in MAGIC swab kit and subjected to automated RNA extraction using the MiRXES MAGIC 32 viral extraction kit. Fortitude 2.1 was also tested using both elite and affordable brands of consumables and RNA extraction kits to simulate the clinical lab settings in resource-limited countries. Our team went beyond the required 300 positive and 500 negative cases by retesting RNA extracts and nasopharyngeal swabs from other accredited labs. In fact, this clinical validation used 3 reference or comparator kits. Among the lessons I learned from this clinical validation is that principal investigators of COVID-19 clinical trials should possess flexibility because the pandemic presents unforeseen changes and diversity of situations. Thus, to adjust easily to constraints and to make fast decisions and plans during ambiguous situations. One has to be attentive to patterns and be daring enough to approach problems in novel ways. Moreover, burnout is real. Taking short breaks to reset stress levels and stepping back a bit could clear the mind and increase productivity. When freedom and the uncertainties in research are essential, it would be wise to be open to the funder about the situation in the ground. Thanks to the wonderful people of MiRXES and their local partner for an environment with open communication and trust. And given the urgency of the matter, a bureaucratic system is the last thing we needed. I have led several government funded projects before, and one of our biggest hurdles then would be the timely release of the budget. Somehow, this concern, this was the list of my concerns. I'll repeat this slide, sorry. While freedom and flexibility to handle uncertainties in research are essential, it would be wise to be open to the founder about the situation in the ground. Thanks to the wonderful people of MiRXES and their local partner for an environment with open communication and trust. And given the urgency of the matter, a bureaucratic system is the last thing we needed. I have led several government-funded projects before, and one of our biggest hurdles then would be the timely release of the budget. Somehow, this was the list of my concerns since an accelerated funding system was already in place. Networking is indeed central to being a principal investigator. And being part of the academe truly has its advantages, especially when I implemented this clinical trial. When I was faced with all these challenges related to patient recruitment, I started calling former and current students to connect me to key persons in COVID-19 control in the country. And that's how I started to connect with stakeholders. In the early stages of the project, I had to meet people face to face to establish strong relationships and networks. As the study progressed, I learned to be more sensitive to the needs of the community and its leaders. This clinical validation has indeed helped increase our research capacity and facilitated the creation of a pool of researchers and technical team that can be redeployed if ever we are faced with another surge or pandemic. To date, the Fortitude kit has been deployed in 13 Singapore hospitals and labs, public and private, and more than 40 countries worldwide. We feel honored to do the clinical validation of Fortitude, which is among the earliest COVID-19 tests. Before I end my talk, I would like to acknowledge the members of my team, starting with my co-PIs, my admin assistants, and the molecular analysts. My sincere thanks to the hard work, sacrifices, and bravery of the swab team and the drivers. Also, thanks to my undergraduates. So I just my sincere thanks to the hard work, sacrifices, and bravery of the swab team and drivers. The project's social media account was maintained by my undergraduate students. No further delays were experienced when the original swab team and drivers got infected, and that's thanks to these healthcare workers who continued with the work until its completion. All images used in this presentation were taken by the group during the project implementation. Otherwise, they have been derived from the following websites. Thank you very much for listening.
Okay, so on the assumption people can hear me, thank you very much for that. Actually, time is up, so we can't take questions, but I'd just like to make a comment that I really understand all those issues you were talking about, all the problems, the bureaucracy, you have to fight, other people just get in the way. We have pre-approved ethical approval. So I have pre-approval to use samples to validate necessary assays to help the hospital do its work in the event of any outbreak. So I have pre-approval. I have to update it to put new people on it, but I actually have pre-approval. So those are really interesting things to hear. Obviously, the same problems are similar in different countries. Can I ask you one question? Did you inactivate the samples at all, or did your staff have to wear pretty intense PPE when they're processing the samples?
we inactivated them.
How did you do that?
That was done by the molecular analyst. We have a separate lab for that.
Okay.
We had, yes. So it's a DOH accredited facility. So yeah, like they just follow whatever protocols being implemented by the DOH here.
Yeah, because our staff they didn't have to wear the pretty aggressive
yes.
High efficiency filtered masks, which are dreadful to wear. So I think that was really helpful. Anyway, I've been told I have to close the session because it's 9:15. So thank you so much. That was really interesting. And obviously we all struggle, whichever country we're in. So well done. Thank you to people for listening, and hope you enjoy the rest of the meeting.